The TNT systems can be used to study protein-DNA interactions in applications such as gel shift assays. In this example, a cloned gene is expressed in vitro using a TNT system to produce the protein of interest. The 5' end of a DNA fragment or oligonucleotide containing the putative protein binding site is labeled with P32 using T4 polynucleotide kinase. The protein of interest and the labeled oligo are mixed and the complexes are analyzed on a non-denaturing gel. First, the cloned gene is transcribed and translated using the TNT system. The cloned gene is transcribed from a T7, T3, or SP6 promoter, and the protein of interest is translated from the RNA. 4 separate binding reactions are typically used in gel shift assays: the oligo alone, protein plus oligo, protein plus a specific competitor oligo, and the protein plus nonspecific competitor oligo. The oligo alone in a control reaction, protein plus oligo. The protein expressed in the TNT reaction is allowed to bind to the labeled oligo containing the putative binding site. Protein plus specific competitor oligo. This reaction tests the ability of an unlabeled oligo to compete for protein binding with the labeled oligo to show binding specificity. Protein and unlabeled oligo are allowed to bind, then labeled oligo is added. Protein plus nonspecific competitor oligo. An unlabeled oligo of similar size and composition to the labeled oligo, but differing in sequence, is incubated with the protein to test for nonspecific binding. Samples are analyzed on a non-denaturing polyacrylamide gel. The oligos bound to the protein will migrate more slowly through the gel than the unbound oligos. Autoradiography reveals the location of the labeled oligo. The oligo alone migrates more rapidly through the gel than the oligo bound to the protein. If binding of the protein to the oligo is specific, unlabeled oligo should block binding of the labeled oligo, causing the labeled oligo to migrate alone. The nonspecific oligo will not block binding of the labeled oligo, and the protein oligo complex will migrate more slowly.